Okay, now we're going to continue with our top 50 books of health and nutrition books of all time. We're on books number 36 through 40. And the first book we're going to talk about is about aluminum. The author's name is Christopher Exley. His nickname is Mr. Aluminum. He's the best, most famous aluminum researcher in the world. It's a very good book. He has some interviews on the internet as well. He sometimes is not as popular as he should be because, anybody who tells the truth often ends up running into problems with the internet. So, anyways, it's an extraordinarily good book. If you're interested in that, that's the book to go to. All right. The next book we'll talk about is Brain Fables. The author's name is Alberto Espay. He's a neurologist. And he was studying Parkinson's disease, Alzheimer's disease, and he came to the conclusion that modern research is mostly a joke. Because they don't understand the diseases well enough, they misclassify the patients, and that leads to the studies being poorly designed and virtually no benefit coming out of it. He says we shouldn't just use the word Parkinson's disease. We should try to determine the mechanism by which they got it, if possible, even the genetics of the patient. And I'll show you some other things he said. He's actually pretty clever. There's some good YouTube videos of him talking. Here's just a little bit about what Parkinson's is all about. Here's the midbrain. It's the top of the brainstem. So in the brainstem, this is the midbrain, this is the pons, this is the medulla, and here's the cervical spinal cord down here. Within the midbrain, when you look at it on cross-sectional, it looks like a Mickey Mouse head, like these are Mickey Mouse ears. And at the base of the ears is something called the substantia nigra, which means like substance black stuff. Okay? Theoretically, on a brain MRI, you can see a swallowtail on the back of the substantia nigra on the SWI sequence, susceptibility weighted imaging, but in reality, quite often the images at this level are blurred and you can't really tell. Okay, there's other tests to look for Parkinson's disease, like a nuclear medicine scan, a DAT scan, so to speak, but nobody really cares for our purposes. Anyways, you need to know substantia nigra loses dopamine neurons, and that's what causes Parkinson's disease. That's what you need to know. Okay, a couple quotes from Alberto Espay. He says, Parkinson's, it's not a prion disease, and it's not a proteinopathy. Okay? He says that Parkinson's disease will have Lewy bodies in the brain at autopsy quite often, but Alzheimer's patients also have enough Lewy bodies to meet criteria for a Parkinson's diagnosis. And I like this because he's getting at the point that autopsy is not as accurate as people think it is. Normal people often have Lewy bodies at autopsy. Non-demented people over 90 have more beta amyloid protein at autopsy. Okay, those with ApoE2, the so-called good gene, will have more beta amyloid and more neurofibrillary tangles than those with the ApoE4, the so-called bad gene for Alzheimer's. Okay, and so what he's doing here is he's kind of showing a lot of the accepted knowledge, so to speak, on Alzheimer's is a joke. Okay. Parkinson's disease patients often have beta amyloid protein at autopsy. About 80% of them do, whether or not they're demented. And those with more beta amyloid protein and more tau neurofibrillary tangles, they live longer. So he's basically, again, saying that the whole beta amyloid theory and autopsy diagnosis of it is kind of a big joke because the patients don't fit the so-called pattern that's in all the books. And then he says, maybe beta amyloid protein, neurofibrillary tangles, and Lewy bodies are part of a protective process, okay? So what he's basically saying is if you see a fireman at a fire, it doesn't mean the fireman caused the fire. Maybe they're trying to prevent the fire or stop the fire. Okay, so, are these protein inclusions like, senile plaques, neurofibrillary tangles, and the Lewy bodies, are these tombstones? Are these the body's response to try to prevent the disease? It's not well understood. Okay. Espay continues. Normal people often have beta amyloid protein at autopsy. Okay. A demented person might not have any of these proteins. So, are Lewy bodies causative of Parkinson's disease? Are beta amyloid proteins causative of Alzheimer's disease? Or maybe they're just tombstones, something that occurs after the fact. Okay, so that's what I'm trying to get at, was the way that Alzheimer's is largely a joke. And you have to get that through your head first in order to start making sense, because the average doctor has been trained that most common cause of dementia is Alzheimer's dementia, and the autopsy findings are pathognomonic, extracellular senile plaques, intrafibrillar neurofibrillary tangles, but that's BS. It's not that reliable, even at autopsy. So you can't diagnose Alzheimer's by historical finding. You can't ask the patient a question. You can't get it by physical exam. You can't put a stethoscope on them and figure out they got Alzheimer's. You can't tell reliably by a blood test, so they can't diagnose it that way. You can't diagnose it reliably by brain MRI, the so-called pattern of medial temporal lobe and parietal focal atrophy. Just you seldom see that pattern. It's not reliable. And then what Espay is pointing out here is you can't even tell at autopsy. Okay, and that's been pointed out by others, including Delatorre and another guy, I forget his name, Francisco, Hispanic guy that did some research with Delatorre. Okay. Okay, now here's the big one that you really should know. This is Jack Delatorre, PhD. This is a great book called Alzheimer's Turning Point. If you're interested in dementia, this is one of the best books ever written on dementia. It's a fantastic book. Okay, so just real quick, a little bit about atherosclerosis. This is so-called Western atherosclerosis, especially, from high-fat diets, especially plugging up the coronary arteries. Also, the pudendal arteries down below to the Johnson is associated with impotence. The pudendal arteries down below to the Johnson are about 1.5 millimeters diameter. Coronaries are about 3 millimeters diameter. So the Johnson almost goes first. They often have impotence before they'll have symptomatic coronary artery disease. The internal carotid artery going up to the brain is about 6 millimeters in diameter, often a little less than that, but about 6 millimeters. And the point is, these are all affected quite a bit by high-fat diets. Now, when you talk about intracranial atherosclerosis, that's also called Asian atherosclerosis, like the Japan pattern, for example, because they would eat tons of sodium, 12 to 14 grams a day, plus they were smoking a lot of cigarettes, a stimulant that would cause a lot of hypertension, but they ate a very low-fat diet, and they also ate some vegetables that were a little bit protective. For this reason, though, just the isolated hypertension, they would get intracranial atherosclerosis, and that's associated with a higher risk of strokes, and that was sort of a characteristic pattern. So that's Asian atherosclerosis versus Western atherosclerosis. Okay, so now we're going to get into the key point about Delatorre's findings. So what that Jack Delatorre did was he would tie off the internal carotid artery high in the neck of a mouse, and then 2 months later, the mouse would become demented. When he first did an autopsy on the mouth, he expected to see a big stroke on the same side where he tied off the carotid, but he didn't see a big stroke. He saw an atrophic brain on the same side. And he did variations on it. He tied off both carotids. He tied off the little arteries in the back called the vertebral arteries. He tied off intracranial arteries, the middle cerebral artery. And he kept on getting the same basic pattern, which was that chronic cerebral hypoperfusion, meaning lack of blood flow, led to gradual death of the brain cells, the neurons in that area. Okay, and so when people first learn about it, they're like, okay, that's interesting, but so what? Not that many people have internal carotid artery occlusions or severe stenosis. However, let's talk about what else happens. The point was chronic cerebral hypoperfusion, undersupply of blood, meaning undersupply of oxygen and glucose. Well, guess what? Atrial fibrillation patients, they lose that 25% atrial kick for filling, so they can have chronic cerebral hypoperfusion, can end up demented. Congestive heart failure means decreased ability of the heart to pump blood. They can end up being mouse equivalents. I call these mouse equivalents. Overtreated hypertension. If you drop the blood pressure too low, because the reason you got blood pressure is to get blood to the top of the brain. If you drop it too low, chronic cerebral hypoperfusion. Aortic stenosis is the aortic valve, in the heart, aortic valve stenosis or regurgitation. After coronary artery bypass grafts, usually called CABG, they run the patient hypotensive quite often, for over a day, so they don't bleed at the anastomosis. That can cause cerebral hypoperfusion. That's not a chronic thing, that's a temporary thing, but there is such a name for it. It's called pump head. Significant number of patients have some cognitive impairment after going for open heart surgery, bypass of the coronary arteries. Severe anemia can decrease oxygen delivery to the brain if it's quite severe. Okay. There's other conditions, not exactly the same, related to like this oxygen glucose delivery, but obstructive sleep apnea, hypoxia. A lot of times that's a fat person, often diabetic. They're getting less oxygen to the brain overnight. I've seen OSA patients, drop into the 60s on their O2 sats. Diabetes can be hypoglycemia, can cause hypoglycemia, less glucose going to the brain cells at night. Often they give themselves too much insulin in the evening. Okay, we're going to talk a little more about atherosclerosis in the arteries of the brain. So here'd be a relatively healthy circle of Willis. COW means circle of Willis. Those are the arteries at the base of the brain, the big, large, dominant arteries that supply blood to the brain. If they're open like these are pretty good. This stuff here that's kind of yellowish looks a little bit like fat. This is atherosclerotic plaque. And these arteries are all narrowed. So instead of having this wide open artery, something like this, you'll have a narrowed artery, a little pinhole here, pretty small diameter there. That's going to decrease the amount of blood supply to the brain. Okay, we need to talk about hypertension because it's super important. Hypertension is the most common cause of little silent strokes in the brain. Diabetes is up there as well. So here's a normal blood vessel, a capillary. The red blood cells are about 7 microns in diameter. The capillary is smaller. It's about 5 microns in diameter. You need to know that, by the way. RBCs are 7 microns in diameter. Capillaries are 5 microns in diameter. You have to know that. That's going to come up all the time. Okay, so because the RBC is bigger than the capillary, it has to fold back on itself a little bit as it passes through the capillary. The little blue circles are the oxygen being delivered from the red blood cell to the neuron, the brain cell. The oxygen has to pass through the capillary basement membrane, this yellow stuff. Then it has to pass through the vascular smooth muscle, the blood-brain barrier. Okay. Then it diffuses to the cell and provides oxygen that the cell will use, like its mitochondria will use that to make energy, to make ATP. All right, well, chronic diabetes, this basement membrane gets thickened. Like you get advanced glycation end products deposited on it, and it becomes thick. So you get a thickened yellow basement membrane. That's going to decrease oxygen delivery to the tissues. You'll also get hypertrophy of the vascular smooth muscles. You get thickening of the collagen in these areas, collagen fibrous tissue, scar tissue, if you will, from the injury caused by the inflammation of diabetes and the physical trauma of the hypertension. Okay, a thicker vascular wall means less oxygen is able to reach all the way to the neuron and diffuse to it. So I made fewer blue circles on this part of the diagram. This is why you want to get your act together. The sooner the better with hypertension and diabetes. Otherwise, you're going to be chronically depriving your brain of optimal glucose and oxygen delivery. So here's the internal carotid artery coming up to the brain, then it bifurcates. There's an anterior cerebral artery, which I don't draw on here because it makes things too complicated. Here's a middle cerebral artery, and it gives off these little branches to the basal ganglia. These are called the deep gray matter nuclei. They're gray, just kind of like the outer cortex, the outer cortical ribbon. That's where the gray matter is. The cell bodies is gray matter. Okay, these little arteries are called lenticular striae. Very common spot for hypertension infarcts. They'll shear off the base of these things, and you get these little lacunar infarcts. Lacune means hole. So you see those all the time. I see those every single day. Okay, now here's the middle cerebral artery. It comes up around the brain, and this is called the convexity because it's outwardly convex. This is the cerebral cortex, this little gray matter ribbon. Okay, that's where the cell bodies are, the neurons, and then their axons pass through what's called the white matter all the way through here, and they go down to the brainstem or those synapses on the basal ganglia. All right, well, anyways, you will often, if you drop your pressure too low, these are sort of end arteries up here going into the upper cerebral cortex, these penetrating arteries, and there's not good collateral blood flow. So you'll often get little silent strokes. These are the cerebral ventricles that contain cerebral spinal fluid outlined in blue. And so lesions adjacent to the ventricles are called periventricular lesions. I see these all the time. I see hundreds, thousands of them every day. They're so common, silent strokes in this area. And usually they don't cause much symptoms, but they start to add up and then they cause a lot of symptoms. Okay, here's a typical normal-looking brain MRI. This patient has normal-sized ventricles. They're relatively small, and the area around them is clean. There's no strokes. Okay, here would be like a typical-looking 80-year-old brain, and I would describe this as mild enlargement of the ventricle, so I call that mild ventriculomegaly. The abnormal flare hyperintensity, white stuff, the bright stuff on the sequence, this is a flare MRI. That's the name of the sequence, which is basically like a T2 with fluid suppression because the fluid is dark. Okay, so anyways, the stuff touching the ventricles, the frontal horns here, that's called juxta. Juxta means to yoke, to connect something. So juxtaventricular flare hyperintensity. The stuff at the level of these lateral ventricles over here, but not touching them, that's called periventricular. Peri means near. Okay, when you have lesions that are out, farther out near the cerebral cortex, that would be called subcortical. So the way this would be read on an MRI report would be juxtaventricular, periventricular, and subcortical extensive flare hyperintensities suggestive of chronic atherosclerotic ischemic disease. Okay, so this is typical. No one would even look twice. No one would even send a view alert or page the doctor for this. This is so common, so typical, it would be expected in a cognitive decline older person. A lot of people don't have much. Some of them don't have any, and that's what your real goal should be. Most of the brains that I see that are demented, their problem is not all these strokes. They just have a shrunken brain. It's atrophic. Okay, moving on to our next book. This is book number 39. This is the book by Russell Blaylock. It's called Excitotoxins, The Taste That Kills. This came out in 1997. It was a landmark book. It was a great book. He's a neurosurgeon. He's a smart guy. His father had died from Parkinson's disease, so he had read quite extensively on Parkinson's disease, and he was fascinated by the concept of excitotoxins. He had read all about John Olney. John Olney was the researcher who figured out that MSG was a neurotoxin, excitotoxin, back in the 1960s, okay? And Blaylock started noticing interesting things when he read Olney's papers and the other papers on it, that animal babies that are exposed to excessive MSG, they have all kinds of problems, short stature, obese, anger management problems, etc. Then he noticed that it could injure the hypothalamus, and that was thought that it made it hard for them to regulate their appetite, predisposing them to be obese. This guy, Mark Mattson, he's a good researcher on glutamate, but glutamate's important for babies' development. These amino acids that are stimulating neurotransmitters are called EAA, for excitatory amino acids. So glutamate's the most important one. That's what you see in MSG, monosodium glutamate. MFG means manufactured free glutamate, and that just means you take a regular protein, like let's say from corn or from gluten or from soy, and you separate the individual amino acids, and then you just add the glutamates to the food. Then there'll be a lot of glutamate that's free in the food. By free, we mean it's not bound to other amino acids, and the fact that it's not bound means that it can be taken up individually by our taste receptors for glutamate, the umami taste receptors in our mouth and throughout our intestinal tract, and it excites them. So we like the food because our ancestors worried about starving to death, and finding a good protein source was useful to them. It's a lot better than starving to death. So you get reward neurotransmitters in the brain released from that, and that helps make people addicted to the food. Cysteine is another protein that is an excitatory amino acid and activates the NMDA receptor. NMDA is the name of the most important receptor for glutamate. 90% of brain neurotransmitters are glutamate, and it's excitatory. Okay. Only about 5% are GABA, that's inhibitory. And then all the famous neurotransmitters, in addition, they're all about in the ballpark of 1%. That's things like acetylcholine, serotonin, dopamine, norepinephrine. Okay, glutamate is often extracted from plants. Blaylock says sudden big doses in excitotoxins is associated with causation for ALS. This is one of the things I've talked about, is that when you have a sudden brain injury from a stroke or from traumatic brain injury, you get opening partially of the blood-brain barrier, and you also get simultaneous opening of the gut barrier, leaky gut. So that creates a perfect storm for a toxin from the gut to get through the leaky gut, then to get into the blood, then to get across the blood-brain barrier, which is another reason why post-neurologic event patients should make sure to eat enough dietary fiber. I'm the one who taught my neurology friends about that, because they don't know anything about nutrition. No doctors do. Okay, this is a neuron. Here's the cell body up here and here's the mitochondria. Here's all the dendrites feeding in information. So this is the reception, the sensory part of it to receive information. Then this is the axon hillock, the beginning of the axon. This is where action potentials typically begin. And the action potential spreads down the axon until it gets to the axon terminal. It's a depolarization, changing the charge across the plasma membrane, so it'll activate these channels here. These are called voltage-gated calcium channels. You can see calcium coming into the cytoplasm. Calcium is the aristocrat in neurons, so when it comes in, It'll cause the neurotransmitter vesicle to travel to the synaptic cleft, to the plasma membrane here, to merge with it, release these neurotransmitters into the cleft. They diffuse across it, bind to the postsynaptic neuron, and exert an effect. Okay, now here is a glutamate synapse in the brain. So let's say the calcium has come in and it's caused increased cytoplasmic calcium. That will lead to the glutamate vesicles traveling to the plasma membrane, and then the glutamate will diffuse across the plasma membrane. It binds to these guys, the AMPA receptors, lets sodium in, that depolarizes the neuron, and then that depolarization activates the NMDA receptor. So here's NMDA receptor. That is the key receptor to know about for glutamate. So when glutamate, indicated here by GLU, binds to it, it opens up and calcium comes into the cell. Some things that you want to know about is that glycine will also bind to this, and glycine phosphate from that herbicide that's sprayed on GMO soy can also activate the NMDA receptor, let more calcium in. Overactivation of NMDA receptor, letting too much calcium into the cell cytoplasm, is a major mechanism for causing excitotoxicity. Excitotoxicity is overstimulation of the neuron. Okay, another point that I would make is excessive aspartame, like from aspartate from aspartame, will also cause increased activation of the NMDA receptor, increased calcium get into this neuron. Overactivation of this neuron can activate a pathway that leads to destruction of the neuron. Okay, this has the perfect name, calpain. Cal because it's activated by calcium. Pain because it has painful consequences. It destroys the postsynaptic neuron and leads to death, apoptosis, programmed cell death, gradual death. So it's bad. If you note carefully, you got magnesium here in the center of the NMDA receptor. Magnesium is protective. So eating adequate amounts of dietary magnesium, which comes from plants, magnesium's in the center of chlorophyll in a plant, it helps to protect the NMDA receptor, make it less easily opened. So it's only opened when it's supposed to be opened. And that helps protect you from calcium excitotoxicity. Here's just a couple of papers. D-aspartate, the aspartate from aspartic acid from glutamate, from aspartame, it'll enhance NMDA receptor responses. Okay, here's aspartame. It can lead through a couple different mechanisms. It can activate the apoptosis pathway. Okay, now we're on book number 40, the last book for this lecture, and this is Fat, Stressed, and Sick by Catherine Reed. By the way, almost every single one of these lectures I show you, there's going to be YouTube videos or somewhere else on the internet videos. So if you want, you can watch all of those. You find them helpful. She's got a really nice talk, Catherine Reed, TED Talk. So Fat, Stressed, and Sick, and it's especially about MSG and MFG, manufactured-free glutamate. And the big motivator for this whole book was that her daughter was diagnosed with autism. And then what she did, this lady, she's a protein biochemist, is she removed all the glutamate that she could from the child's diet, and her kid got better from mild autism. I saw the videos of it. It's for real. She's for real. She's been on a bunch of internet and other interview shows. The food companies know that people love glutamate. They know they love salt, sodium, and they love high fructose corn syrup, and they want to get just the right mouth texture. So they do all these research studies where they bring in all these regular people and have them taste test their different foods. And what they're trying to do is get just the right amount of MSG, salt, mouthfeel texture, fat, high fructose corn syrup, so that the person hits a so-called bliss point, where they like the food so much they always want more. Like the old bet you can't stop with just one. Okay, so what some of the things Catherine Reed figured out as a PhD biochemist was the more you process a protein, ultra-pasteurization, some type of enzyme lysis, extract it in some way, hydrolyze it in some way, separate the fat out, ferment it in some way, the more you do all this stuff, the more you free up the individual Gs. The individual Gs are glutamates, the glutamate residue. So these glutamates freed up are now called manufactured free glutamates. So it's manufactured because you had to do this manufacturing process. Free because they're separate. They're not bound to any other amino acids. Glutamates. They're glutamates. And that's all the body cares about. It doesn't care about the monosodium in MSG. It just cares about the glutamate because glutamate is the excitatory neurotransmitter. And there's areas of the brain, even in an optimally healthy person, that lack a blood-brain barrier where you have chemoreceptors like the circumventricular organs, the area postrema vomiting center, etc. Okay, so here she is. This is Catherine Reed. She got a really good TED Talk on MSG, manufacturer-free glutamate. Her website is unblindmymind.org, and it's quite impressive how her daughter improved. There's probably more than one mechanism of causing autism, but it certainly seems that in some patients they can respond great to removing MSG from the diet. Her daughter was cured. So she had to go to great lengths to learn all the different ways they hide MSG because they know the companies know that the public doesn't want it. So at first they'll hide it under a bunch of different names, but nowadays the big thing they do is they just make it free. So they just chop up proteins that are high in their concentrations of glutamate, and they free up the individual glutamate residues. And again, all that matters is how much glutamate there is. That's what the big deal is. And we love glutamate. We can't help it. We're like a cat to turnip. We are programmed to seek glutamate. Our reward systems of our body are made for it. Let's see, what else? They make the MFG, manufactured free glutamate, from corn or from gluten or casein, whey, or soy. She says that excessive amounts of free glutamate will cause an increased risk of obesity, diabetes, hypertension, autism, ADHD, anxiety, dementia, or cancer. So it'll cause obesity because it tastes good, and people eat more. You put salt, sugar, fat, MSG on a food, people will eat more of it. It can cause ADHD, I would expect, and anxiety because it overstimulates the neurons, so that can cause anxiety. MSG is fed to rodents to make them obese for research study. It's a flavor enhancer. I knew a neurologist who had done research on MSG, and he told me the main purpose of it is just to make stuff taste good so people buy it. John Olney's the guy who did the research in the 1960s who showed that it's a neurotoxic excitotoxin, overstimulates neurons to the point where they can die. The companies hid it for years with all kinds of names like barley malt extract. So whenever you see extract, or malt, you should think of MSG. Main dietary sources are all these ultra-processed foods, like with a whole bunch of stuff on the label. I only eat foods that have one ingredient. This is rice, this is beans, this is quinoa. Okay, the big food is a $1 trillion industry, so it'll pay to have fake research to say MSG is okay because it knows that it needs it in there, MSG or MFG, to get the people to like the food. Avoid all processed food. It's all full of this stuff. Avoid also mouthwash, chewing gum. Never eat protein powders. The only processed food I would eat is single ingredient plain oatmeal or quinoa. Okay, well that's it. I hope that was helpful.